The second speaker of this morning's session is Thomas Carlson. We overlapped just very briefly when he was at Harvard working with Patrick Cavanaugh. So welcome, second speaker. Welcome.
Good. I think it's working. I'd also like to thank the organizers for the invitation. It's wonderful to come to Italy and attend this conference. Oddly enough, the last 2 years I wanted to come to this conference. The 1 year I forgot. Last year I tried to register and I was locked out. It was just too late. There's too high demand to get here. So it was pretty much instant response when Marius emailed me. It's like, would you like to come and give a talk at the conference? So the first talk of the morning actually, in many ways, kind of foreshadowed some of the things that I want to talk about. Of course, there was a discussion of Jim Haxby's kind of seminal decoding study, which has an interesting history for me because my first decoding study was actually just a reanalysis of Jim Haxby's data, and that's how I got started. And interesting enough, I don't know if you remember, that's when I met Alex Martin at my poster, and I was terrified. He made me really nervous. It still makes me a little nervous, but not so much anymore. Oh, good. Okay, so today what I want to talk about is kind of a shift in thinking in my lab that I'm trying to put out there more to the broader academic community, because I think it's maybe a shift that we need to make in thinking about decoding research. So there's, all these kind of devices here represent information. A computer has a hard drive, Apple iPhone or your smartphone, many of us have in our pockets, and that information is encrypted sometimes really well, as the example of the iPhone in the recent history of the US, the Canon, but also things like as simple as a chalkboard represent information, and kind of painting and art, all are forms of means of communicating information. But there's a kind of fundamental division there in that on one side, the decoder is sort of part of the machinery. An engineer is sort of figuring out how to kind of decode things from the hard drive and read it out and call it up for your apps and such. But on the other side, we need a human observer. So if we have a chalkboard, the encoder might be a professor or something, or a painter, but we have to be observers and we become the decoders. Our brains become the decoders. So the interesting thing that my lab's been kind of thinking about a lot is sort of, and on this side, the brain is also its own decoder. The brain clearly represents information, but somehow it has to take that information from the representations for making behavioral choices. So I kind of refer to this as the problem of egocentric decoder. I'm not referring to myself, but as kind of neuroscientists, we sort of really want to just dive in and we just want to find that information in the brain. Is it represented in V1? Is it represented in LO? Is it represented in IT? We do kind of thinking like maybe we'll sort of adjudicate between areas and say, well, this information is not represented in V1, but it is represented in inferior temporal cortex and inferior parietal cortex. So those two areas must be somehow participating in the representation that the brain is using for behavior. And in a really very nice paper that came out here by DeWitt, I just draw a quote, and he's sort of making this distinction as well, so we need to change our thinking a little bit about what decoding actually means. He says, one can simply not measure a physical property, for example, the flow of electrons in a wire, or firing a neuron, pattern activity measured with fMRI, and assume that we've actually measured information. Rather, what we need to do is construct models of how that activity can be used by the receiver. And DeWitt's point was this, is we often think of ourselves as neuroscientists as the receivers when we run our studies. But what we really want to be thinking about is how the brain is acting as a receiver. And then we can actually figure out whether the brain is using that information. So it turns out that this is actually not a new idea. In fact, it's come up many a times, and it's starting to come up more and more in the recent literature. But Fechner, in kind of one of the fathers of modern psychophysics, was thinking about these things, and he actually made this kind of separation between what he called inner psychophysics and outer psychophysics. So outer psychophysics is what kind of most of us do when we do behavioral studies in the lab. We kind of measure reaction times or accuracy, whatever measure that we do, and then from that behavior, we try to infer something about mental representations in the mind. Inner psychophysics is actually thinking about how the quantities are represented in the mind, and then how they kind of relate to behavior, which is basically kind of what I want to talk about today, and that's sort of the brain operating as its own decoder. Sadly, but not surprisingly enough, Fechner just didn't have the tools that we have at our disposal today, so he kind of was limited to kind of classical outer psychophysics, which has actually brought us a long way. So cognitive neuroscience is kind of the modern manifestation of Fechner's inner psychophysics. We now have a bunch of different methods that we can sort of measure brain representations and measure information in the brain. But as I said, our ambition is kind of extend that further and to sort of think about and describe models of actually how processes occur in the brain, how they're neurally implemented, and characterize how information is encoded. That's more on the representation side. But most important, or more interesting to me, is the decoding of itself of information by the brain. So in a recent kind of theoretical paper, he was never really my student, but he just hung around a lot, and we did a lot of work together. Brendan Ritchie, we kind of started thinking about kind of driving a wedge between what kind of decoders tend to think about and kind of our ambition. And we call this the distinction between biological and psychological plausibility. So biological plausibility is things like classic decoding. We tend to use what they call linear decoders because we can easily see how the brain could implement that neurally. In fact, DiCarlo has actually argued things for kind of objects. One of the main functions of the ventral temporal pathway is to transform early visual inputs so that they're linearly separable in inferior temporal cortex. So, from DiCarlo's perspective, really, we just want to kind of represent information in a way that can be sort of read out by a linear classifier. But if we want to go and talk more about kind of Fechner's inner psychophysics, we have to talk about kind of how the structure of these representations actually relate to behavior. And this came up as one of the really late questions in the previous talk. I think somebody was talking about sort of how information, two types of information, they're represented differently, and one seemed to be more structured towards a behavior and the other was not, and difference between blind subjects and sighted subjects. And there has actually been, people have been thinking about this a little bit in some kind of ways. So I think one of the first studies that kind of got at this was a study by Mark Williams. And the study was basically contrasted decoding objects that were consciously perceived versus objects that were not consciously perceived. And what this study showed was, didn't matter whether the subject consciously perceived the object or not, you could decode the information in V1 cortex. But in LOC, it was dependent on whether or not the subject consciously perceived the object. So here's just kind of one example. We're trying to link decodability, and it's actually kind of a separation where just because we can decode doesn't necessarily mean that the brain's using that information. The implications of Mark's study is that the V1 information is always represented even when it's not consciously accessed. And I think also kind of Kriegeskort's recent kind of representational similarity analysis approach, some of the studies kind of marry with this also. where they take some sort of behavioral measurement, for example, judging the visual similarity of objects and compare that to neural similarity. And we've done some work in that area as well. And I think that's also kind of a promising approach. We're actually trying to connect how the structure of brain representations kind of relate to our own behavior. But really, what we're trying to do is take it to the next step and actually try to explicitly develop kind of models of how that information can be read out. Okay, so here's a general outline for the work I'm going to talk about today. First, I'm going to very briefly go through, I'm sure almost all of you are familiar with decoding research, so I'm going to just do 2 minutes on how we can measure the structure of information using decoding research. And then I'll talk about how we can actually predict behavioral outcomes using one, what we call psychologically and biologically plausible model. And then I'm going to discuss some alternative models and the whole range of massive issues associated with trying to develop a model of readout of how we take information out of a brain area. And I'll warn you in advance, it's going to kind of go from very concrete to very messy in terms of the data. I don't mind messy data because when we're sort of working on this last bit, which is still work in progress, we learned a lot about how hard it is to actually develop a model. So in that last section, what I want to kind of convey to you is kind of all the limitations and all the things we don't know for kind of constructing a model, which is something that actually seems pretty simple. We just sort of need to read information from representation. It doesn't seem that hard, but it turns out it's actually extremely difficult. Okay, so we'll start with measuring the structure of information. We'll give 1 minute on this. Perhaps we can maybe even call James up to do this. But the idea with kind of classic decoding research is we're going to measure brain activity using one of many different types of neural measurement, largely developed in the domain of fMRI. You're seeing more research now with MEG and EEG and single unit recordings, basically anything that kind of gives you a measurement of a representation in the brain. From there, we take pattern classifiers. They're going to learn certain patterns of activity that are associated with stimuli, categories, tasks. So that's kind of graphically depicted here. We might show a person a whole bunch of different images of cats. And if we have this little 9 voxel image, that would be sort of the response, color-coded response to cats. We'll show the subject a bunch of pictures of little dogs and puppies, and that would be the response to a bunch of different exemplars from puppies. And then if we kind of project that into kind of voxel space or activity space, which is sort of a concept that's important to think about, because we'll be talking a lot about spaces here in the talk. All the cats kind of generate a high amount of activity in voxel 1 and relatively low on voxel 2, and high response on voxel 2 and low response on voxel 1 for the category of dogs. And then, of course, we withhold some of our data, and then we're going to present that data. And then if we maybe present stimulus B or stimulus A here, it lands on here, then we're going to classify that a dog, and then we're going to basically test our classifier. Okay, and that's just the simple process of kind of decoding. So we're going to use this to decode novel measurements or stimuli. Okay, and then I'll jump right into work that I'm sure most of you are familiar with. This was the 2008 study from Kriegeskort. So now just doing cats and dogs, we're going to do, Nico did 92 different exemplars, and we're going to measure the pairwise decodability for all possible combinations of those exemplars. That gives us something called a dissimilarity matrix, and then just for kind of simplified pictorial reasons, we can flatten this dissimilarity matrix and get a rough idea of kind of what ITC cares about or what the representation looks like. So in this figure, basically distance is going to equal decodability, and you can see that all the faces are kind of clustered close to one another, which means that it's hard for us to decode different faces from one another based on the patterns of activity in ITC cortex. On the other hand, something like a face and a kiwi, those are kind of a long ways away from each other, so it's relatively easy for us to decode those two objects. And from this, you can sort of get some intuitive ideas about the structure of information in ITC, and the most notable and kind of one of the dominant separations being the distinction between animate and inanimate objects. So if we draw a line here, pretty much everything on this side is animate, and everything on that side is inanimate. Of course, in my old work, we sort of extended this to what we call time-varying decoding analysis or sliding window decoding analysis using MEG. And now instead of a kind of static picture of the representation, we can actually see how the representation emerges and evolves over time. So hopefully this movie will work, and I'll try to pause it at a couple of key points. So sorry, I should pause it here. So up here on the top is the time. So right now it says minus 20 milliseconds, which means the stimulus actually hasn't been shown to the observer. And that dot in the middle is actually a clustered mess of objects, but they're all sitting on top of each other because we can't decode them, obviously because the stimulus is not being represented, it hasn't been shown to the observer. So if we continue on here, let's say it's actually, I won't go back to, but about 60 milliseconds, you see them sort of expand out from one another. And that's indicating now we can actually start to decode different exemplars of objects. And it's also nice because there's kind of correspondence in terms of physiology. We know it takes roughly about 40 to 60 milliseconds for information to go from the retina to early visual cortex. So in some sense, you can sort of think about these kind of around 60 milliseconds as an early visual representation of the stimulus. And you don't really see a lot of kind of categorical structure yet. We'll go a couple more frames to, let's see, 120 milliseconds. Now you see a couple of things. The faces, the three faces are sort of off on their own in their own little cluster. And you can think of sort of this is the brain's kind of early construct of a representation of a face. And there is starting to be some semblance of a boundary between animate and inanimate objects. We just go a couple more frames. We see that nice animate-inanimate distinction that we saw in the Kriegeskort study. So about 160 milliseconds, so we kind of have that nice animate-inanimate distinction. And of course, since we use MEG, this is kind of nice because now we can sort of measure kind of these dynamic emergence of object representations. Okay. So just up to this point, basically I told you all old hat, old news, and basically how we can actually measure the structure of representations and get to, I think, what Nico calls representational geometry of the structure. And now is the important, kind of more important bit, in my mind, is how we can actually think about this structure and try to use the actual structure representations to get to behavior. Okay. All right, so we know we can use our decoding methods to kind of measure representational geometry, and now we want to build a model of how to read out information from this representation. So the initial starting point, and this is a major assumption of even the RSA framework, is that representational distance is important. How far things are apart in the representation is going to determine behavioral outcomes in some way. And the simplest sort of interpretation of that is basically anything that's close in the representational space is going to be a harder decision for the brain. So if you're trying to discriminate between objects, and importantly, assuming that you are using this representation, if these two objects are very close to one another, for example, the baby and the bearded guy, even though they don't really look that much alike, because they're really close to one another in the representation, that's going to be a hard choice for you to make. On the flip side, maybe take the bearded guy and the tomato, because they're a long ways apart from each other. It's easy for them to be separated by the brain, so it's an easy choice for the observer. So we have a kind of straightforward hypothesis about distance in representational spaces, and we also have a straightforward prediction that we can make about kind of how the geometry of these spaces is going to relate to behavior. Okay, so we kind of took the next step with this, and then we say, okay, let's think about category decisions. And you can even think of it as importing it from a kind of a decoder's perspective. And that's exactly what we were thinking about when we started this study. It was like, well, maybe the brain is sort of acting like a decoder, a linear classifier. And if we take this kind of original object space geometry, we'll go back and sort of, you can see there's the original kind of study of the geometry in IT. I'm just going to change those little dots and such. And we use a classifier to fit some sort of boundary through the representational space. It's going to split these two objects into two categories. Then we can use kind of classical signal detection theory to kind of make predictions about behavior. And now it's not that, for example, if these two faces are close to one another, it's whether or not items are close to this boundary. So the prediction is, for example, here we have a picture of an ear, and it's very close to the boundary in this high dimensional space, and we're going to kind of project it on this discriminant axis, and then because it's close to the boundary, it's going to be a difficult choice. And the same thing, here we go with the face. The face is a long ways from the boundary, so that should be a relatively easy choice. And we can do the same thing on the inanimate side. So again, we have a very straightforward hypothesis that it's actually a boundary kind of bisecting this representational space that's important. And you can think of these as sort of weights on a neuron. And because the way the weights line up, close to a boundary is going to be a harder choice, and further from the boundary is an easy choice. So it just so happened at the time I was moving universities, and we weren't really starting our fMRI program, but I've been good friends with Nico for a long time. So I said, well, forget it, kind of like I did with Jim Haxby's data years ago. I just said, well, why don't you just send me your data and we'll do the study. So Nico kindly just sent us his data, and we immediately started working on this. And it was actually a fantastic study because it was run by two, not even undergraduates, high school students who did the whole study in 2 weeks. Basically, I asked them, well, why don't we go and try this MTurk thing out and see if we can get our behavioral data. By the way, MTurk data is really high quality. It is amazing. So we took the original stimuli from the Kriegers-Court study, and we just had people on MTurk categorize the stimuli as animate or inanimate as quickly and accurately as possible. We go to our prediction. Again, so we have our prediction that we're going to have a negative correlation between RT and the boundary. And basically, we found that it was confirmed very nicely with one caveat. It worked great for animate objects and not at all for inanimate objects. So here we have the reaction times, and here's the distance from the boundary. And you can see the red dots, those are the animate objects. We kind of got a really nice kind of line there, good correlation, almost 0.7. But for the inanimate objects, sort of flat, 0. So we started with a very kind of simple model of readout, and actually made kind of a big step forward. We can now make accurate predictions about future behavior based on this representation. But it's not a perfect model. It doesn't work for the inanimate objects. And we're still sort of thinking about that, and there's a lot of complexities to do with that, about variation and things like that, but we can maybe talk about that in the discussion period. So in this study, we really wanted to go kind of full circle and not just stop at the point where we're sort of mapping reaction times to representational distances. So we wanted to sort of actually kind of connect this with models of human decision making. And this was partly a problem. I shouldn't say partly a problem, but these really effective high school students knocked off the first part of the experiment in 2 weeks. So I told them, well, why don't you try to model the decision process? Because I had them for the whole summer. And then they wrapped that up in about 6 weeks. Montgomery Blair is one of the best schools in the United States. They're really impressive students. Okay, so what we did was we constructed, there's all sorts of variants of decision-making models, and it turns out it doesn't really matter which one we use because we only care about a single parameter, and that's basically representational distance. And representational distance is going to kind of equate to the strength of evidence. So we can't actually adjudicate between models of decision-making, but we can at least show how we can transform representational distances, what we think is important, into an actual behavior. So we picked one of them. It's one that's used by Gold and Shadlin. And basically, it's kind of a form of a drift diffusion model. And the rate of diffusion, sort of the slope of this line, is the strength of the evidence. So basically, all we're doing with this model is if it's close to the boundary, it's going to be a shallow slope. And it's going to take a long time to kind of cross this sort of decision threshold. And if it's a strong evidence farther from the boundary, it's going to be very steep, and it's going to cross that decision threshold. And then we just iteratively run the model. There's some noise added in. You just sort of add Gaussian noise, and then you let the model sort of converge and make actual decisions, which are going to correspond to reaction time. So the more iterations, the longer it takes for the brain to make that decision. From there, we get this really pretty colorful rainbow plot, where the steep ones are things like faces, and here's kind of the diffusion process for all the different other objects. And again, we also get a nice fit with when we do our reaction times with number of iterations. But when we add this extra diffusion model to our data, now we can also explain the variance in the observed reaction times. So we've gone sort of from stimulus to representation to reaction times, mean reaction times on variance. Very nice kind of characterization. I think I'm doing okay for time. I'll briefly touch on an MEG study. I don't want to go into a lot of details with this, but it's really sort of the same idea we do with the representational dynamics. We have a decision boundary with animate objects and inanimate objects. Instead of a static representation measured from inferior temporal cortex, we have a dynamic representation that changes over time. We're measuring using MEG, so stimulus onset, nothing there, it hasn't reached the cortex. 80 milliseconds, about the time it reaches V1, they start to expand, but there's not a lot of organization. And at like 240 milliseconds is the time where peak decoding occurs, and we have good separation between the categories. And you can see this is a plot of the decoding accuracy over time. So at about 0, nothing, and then we shoot up about 40, 60 milliseconds, then we peak about 240 milliseconds, so it drips down. Skip over those details. Two things that we did with this study is we had 2 different tasks. We had subjects either doing active categorization, where we're going to use their reaction times, and that's the animate, inanimate. But also, when they're seeing the images, there's little tiny letters at fixation. We call this a distracted viewing task, and now they're just saying whether that letter was a consonant or vowel. Okay. I'll just jump through, jump. Okay, so basically, we have 3 lines here. It's a little messy. Categorization here, this is when we use the object category reaction time data with the data collected when they were performing the object category task. And you can see right here about 240 milliseconds. We have significant correlation between the reaction times, so we have our link. We're using our model to link it to the behavior. Then we have distracted viewing. This is sort of just a check, because maybe we have we're reading out some motor activity. So what we do is we'll correlate the reaction times for the distracted viewing task with the data from collected when they were doing the categorization task. And that's pretty much flat, not significant. And then we have what we call the crossover, and that's basically when the subjects are performing the passive viewing task, so they're ignoring the stimuli, but then we use their behavioral data for the reaction times. And that approaches significance, but it doesn't quite get there. And the reason we did that basically is because that's pretty much exactly what we did with the fMRI study. In Nico's original study, they were doing a distracted viewing task. And we used kind of our behavioral reaction times data. So even when these object stimuli are being ignored, ITC's representing them in a way with the structure that we can actually predict their reaction time. And that basically means that it's a very automated process. And finally, we found that basically, so the strength of the decodability could be linked to kind of the strength of the RT prediction from the model. So basically, the more decodable the stimuli, the better we could predict reaction times. And the take-home message from this is basically, it seems like the brain the optimal time to read out information is the time when information is most decodable. So it seems like the brain is reading out that information at the kind of optimal point in time. Okay, so just to finish up that section, we call it, so this neural distance to boundary model is what we call both biologically, it's using a linear classifier, and psychologically plausible because we're now using the structure of the representation to actually predict behavioral outcomes. And it's interesting also to note that all the behavioral outcomes we're predicting are often with new sets of subjects, except for in the MEG study. And this is also, already touched on, biologically and psychologically plausible. Okay, so some quick interim conclusions. Representational distances are important. We've replicated our fMRI study with MEG, showing evidence for kind of optimal readout. And we presented one model that was kind of a first step towards Fechner's dreams, where we're looking at kind of fine-grained structure and representations to predict future behavior. Okay, yep. All right, so I think I have maybe 10 or 15 minutes left. And now it'll get a little bit more messy. Apologies for this, but I figured, well, I got a big room full of smart people. Maybe they can help me sort out this mess. Okay, so we've done our work with fMRI and MEG. Now we're basically extending this more models, because there are, the distance to bound is just one form of model. It's from the cognitive literature, and there's a whole bunch of them out there. So we can kind of import more models and look at them. So now we're going to extend this with more models, and I call bigger and better data. Okay. All right, so here's our new data set. This was wonderful. I think it was maybe a year or two ago. I was walking around Society for Neuroscience, and actually I'd been fishing around to get some single unit recording data for years, and I could not find anybody that was willing to share. But then I hit up on this poster at the Society for Neuroscience by a postdoc named David McMahon, where they did probably one of the more ambitious studies that you can imagine, where they first used fMRI to localize a face patch in the anterior fundus of a primate. And then they get recorded from 30 neurons, 10,000 different stimuli from 10 different categories. So that big mushy mess that you can't really see, that's all the stimuli, that's all the 10,000 different stimuli that they showed these monkeys. And this is wonderful because one of the problems, so the limitations when we did our kind of initial fMRI study is it's great, we can get our behavioral predictions, but we only have the kind of static representation of IT, but we know that there's sort of dynamics in the neurons. And when we do MEG, well, some people are stronger proponents for localization in MEG, but you always have to be concerned about that. So we don't really know that we're measuring from a single area in the brain. So when we get to use physiology data, now we know where we are in the brain. We're in the anterior fundus face patch, and we have very excellent temporal resolution in the form of spiking activity. Okay, so, and we have these 10 beautiful categories, which includes human faces which are important because it's a face area. Pictures of human bodies, monkey faces, monkey bodies, birds, butterflies, plants, man-made objects, scenes, and something called Fourier descriptors, which are just sort of almost like control stimuli, which will so only sort of become relevant in a little bit. Okay, so let's kind of take, this is almost like the fMRI version of the picture, if you will. We're going to just look at the kind of a sneak peek of the time average data. So where this is average data for 2,500 of the object exemplars from 0 to 350 milliseconds post-stimulus onset. And they're color-coded by category here. So we can see the red dots are human faces. They're kind of right here. The human bodies, you can see them kind of scattered around here. The big kind of this blue cluster here, those are the monkey faces that you might expect because we're recording from a face area and a primate. Monkey bodies, they're kind of somewhere kind of in betweenish. Sorry, I should point out these are sort of the top 4 principal components from the data. So you can think of these as eigenneurons. That's what Dave likes to refer to it. So even in this sort of just static picture view, you can see things like, of course, naturally, we have this sort of clear separation of monkey faces data. They're getting a very distinct response. You can imagine drawing a classifier and easily being able to decode monkey faces. Okay. But importantly, we want to think about things in terms of tasks. So when we want to think about sort of, when somebody has to make a decision, is it a monkey face or not? And that's what this is we've sort of task coded the data. So now the red dots are target stimuli, which are the monkey faces, and all the blue dots are any other category other than monkey faces. And then there's also this little black dot here with the black circle, and that's just kind of baseline activity when there's no response from the neurons. And it turns out baseline activity is going to become an important problem for the readout in a few slides. Okay, so our first stumbling block we ran into and this is really sort of, we need to think about sort of dynamic representations. So when we're trying to construct this, first thing we say, well, how do we want to think about our decoder? And we kind of think about it as a passive. There's a lot of object recognition is passive. You recognize your friend. You don't look out there, who's that? And you just sort of go, oh, that's my friend Jeff. So if it's a passive decoder, what that means is probably the readout is going to be static. And what I mean by that is if we're going to draw a line through the representational space, that line's not going to move. Where everything that divides animates from inanimates is going to be fixed in that space. Other theories do propose that you could have a dynamic readout, but it makes the models a lot more complex. And I'm not really that convinced about the dynamic readout, at least for object recognition. For some things like motor movements, dynamic processes make a little more sense. Okay, so when we think about the static representation, like our fMRI data, this is what I call a cognitive decision bound. It's like our LDA model. We think about it as something like this. We have activation here, dimension 1 here, dimension 1 here, and dimension 2. We draw a linear classifier, and then we measure the distance, and we make our prediction. Of course, we're using single unit recording data, so we have these kind of dynamics. Before the stimulus, there's not much happening, and then the stimulus, there's a big response, and then it sort of falls off. So really what this means is we have to add this sort of time dimension in considering our models. What are we going to do at that time dimension? And the first thing that became apparent in this is a 2 alternative choice for an observer is actually a 3 alternative choice for the brain. And what I mean by that is if you sort of sit somebody down and you say, is it animate or inanimate, the brain first has to decide, is there a stimulus or not? And then it has to decide what category the target is. And you'll see why that's important when we get to kind of the neural the models in a second. Okay, and we tested a whole bunch of what we call the psychologically plausible models. These are models from the cognitive decision-making literature. They're kind of very big and popular. We have 2 classes. We have what we call decision-bound models, and prototype models. So the decision-bound models are like the ones we've been talking about all along. There's a line dividing the representational space, and we measure distances from that line to determine how difficult the choice is. And then there's a class called prototype models. Here, the line's not so important. It's basically what the kind of ideal exemplar or the prototype exemplar is. And there, the predictions are based on sort of where this prototype is spaced, and then if we want to know about individual exemplars, it's how far they are away from the prototype. So here we have kind of examples using target and non-targets, and we measure kind of distance from a prototype. And you can already think where there's going to be stumbling blocks for doing something like monkey face, non-monkey face, is the distribution of non-targets is going to be kind of a mess. If you remember that earlier plot, they're kind of spread over in a V. So there's not going to be, you can't really compute a lot of statistics for doing prototype choices. So we use 3 decision-bound models, what we call the LB, the linear boundary model. I put an arrow because this is the model that we've been talking about up to this point, is the linear boundary model. And that just is a classifier trained to separate targets from non-targets. Another one called a baseline boundary model, and we implemented this again because baseline activity is going to become an issue. So this classifier discriminates targets from baseline activity, and then we're going to measure distance from that line. And then we have this dual boundary model, which is actually 2 classifiers, and you can have to think of the brain as actually trying to simultaneously read 2 forms of information out of the same representation simultaneously. And that's a question that maybe we can talk about later, is whether the brain actually does that. Okay, and we have 2 prototype models. One's classic prototype, so we find where the kind of prototype for an object is. And that, I should say, for a prototype for us is just the average of the 2,500 exemplars from a particular object. That's how we define our. So we have classic prototype, which uses the distance from the prototype, and we call something called the baseline prototype, which is basically uses baseline activity and then measures how far kind of on this vector going through the prototype something is. So if you have an activation there, it's, let's say 3 units there, but it's very close to the non-target prototype. I know, how am I doing? I think I got about maybe 5 or so minutes. Okay, and we'll stick with the kind of the big important lessons, even the 5 minutes, beautiful. The big kind of important lessons, and so we can dive into the messiness of the data later on. Okay, so the first thing that came out is all of the models can successfully decode all of the categories. And that is sort of important because we're doing things like decoding butterflies, random images of objects from this single unit data. So we know that just establishing whether or not we can decode, that's not good enough. That's not going to help us tell anything about our models because all of the models can decode. So it's not helping us to kind of adjudicate whether one model is better than not. I do put a little asterisk there. The dual boundary model does do a 3-category classification, and that's why it's below chance. So if people's eyes have wandered over here to this plot and looking at, this is the kind of time series data, you can see all the other models are at chance, 50-50, target, non-target, but the dual boundary model's down here, and that's because it's just way how the models are evaluated. We want a common way to evaluate the models, and the dual model makes case punished for that. Okay. The next thing we learned is model guessing is informative. So often in decoding research, when we see a classifier performing at chance, we interpret this as no information. And even DeWitt points this out, as that we should be really careful about making that conclusion because we might be using the wrong classifier. There might be information there, but we just don't know how the information is being read out. So even thinking about chance performance is a little bit tricky. But when we sort of plotted kind of the decoding accuracy separately for the targets and non-targets, what we see is it's getting the non-targets right 90% of the time, even before stimulus onset. and it's getting the targets wrong almost all the time, even for stimulus onset. And the reason that is because the classifier is basically always saying non-target. And it's basically using kind of the ratios to get all of these right and most of those wrong. And why that has to do with sort of this baseline problem and how do we treat baseline activity. So up here we have the kind of data trained. So here when we're doing the monkey face discrimination task, you can see all the reds, all the monkey exemplars, all the blue are the non-monkey face exemplars, and here's our trained classifier boundary, and the circle is the standard deviation, or 3 standard deviations for baseline activity. And basically you see almost all the baseline activity is falling on the non-target side of the boundary, so the model is always going to guess non-target during the baseline. And why that is basically important is because if we assume that we have a brain that it's a passive decoder, which I think is fair because we automatically recognize a lot of things without sort of cognitively trying to make a decision, then this type of decoder would be feeding bad information to these higher order processes. It would consistently be feeding non-target, non-target, non-target. So we have to figure, this again is sort of a reiteration about how we have to think about treat baseline activity. And it turns out 2 of the models, the linear boundary model and the baseline boundary model, both exhibit this behavior. I'm skip through it. So the next thing is to use reaction time data. So now we're talking about using more fine-grained structure to really get at what might be a better model. And unfortunately, the data gets better, but it's still very messy. So what you see up on top is the decision-bound models and these little dots underneath. And don't worry, you don't have to really see the little dots. I'll tell you the important points. So here's the correlation between distance and reaction time behavior. And you see that there's lots of little dots down here, which means our linear boundary model is doing a good job for predicting reaction time for target stimuli. The baseline boundary doesn't really do anything well except for monkey faces, which makes sense because we localize this as a face area in a primate. And the dual boundary model does good for a bunch of other categories. So all our decision-bound models are doing good. Interesting, the classic prototype model really failed at this. We couldn't relate kind of the readout using the prototype model. But this also got us thinking about another problem that we have to think about when we're trying to build these models is, so what is the criterion now? We know that this is a face patch in the primate cortex, so does that mean, well, if a model is successful for doing primate faces, does that mean it's the winner? But we don't actually know if the brain's actually even using this representation. So we need to, in order to really move forward to this readout thing, we actually more firmly establish this question. And we might have to do that with cooling or some other type of study, optogenetics. And these are all for just the target stimuli. And we also did it for non-targets. And again, all of our linear boundary models fare well. Now the classic prototype model doesn't work for predicting reaction time for targets, but it does for non-targets, which is sort of interesting. And again, I reiterate the point, like to really figure out this sort of readout, we actually have to know whether the brain is reading out information and what type of information it's reading out from this. Okay, so I told you, and I promised I would finish on messy data. I think I have delivered. Okay, so we just finished talking about alternative models. That's, of course, something we can discuss. But we don't have any straightforward answers yet on that. But hopefully the idea is we'll kind of work on that in the future. So, of course, I want to acknowledge all the people that have worked on this. Brendan Ritchie is the student who was never really my student, but hung around a lot and did a lot of work with me. He's been working on me with the theory, this kind of theory, and developing why it's sort of important to think about readout. The fMRI study was done with Brendan, the 2 high school students that I mentioned, and with the help of Nico, who provided his fMRI data. The single unit recording data is being done with my student Tyle Grutzwagers. And of course, Dave, a big thanks for David, the 2 Davids from NIH for providing their single unit data. and of course the Australian government. I'll finish there.
That was really great. So this framing that you have is really interesting, and I have two I think are interrelated questions. So if you start to think about the brain as reading out its own information, which you're right, it has to be doing, does that mean we have to kind of give up on this localist approach of trying to decode from a particular region? Because presumably the brain has available all of itself, right? And, you know, of course there are practical limitations, you know, especially with neurophysiology, you have to record somewhere. But the question is, you know, how do we practically take that step to start to think about how to classify information across the entire brain without necessarily just dumping the whole brain into a classifier, okay? So that's question one. Let me ask you the second one, then you can respond to both. If I can remember the second one. Well, why don't you answer the first one, and then I'll try to come back to it.
Okay, so my understanding of the question is, should we stop doing fMRI? Well,
should we go beyond trying to think about how to classify information in a particular region? I got my second question. Maybe it'll clarify it.
Well,
so could you just say something about how do you implement this idea of a boundary, right? Because the boundary for one region will be very different than the boundary for another region. And so, for instance, if you think about like animate, inanimate, that might be a relevant boundary early in the visual system. And as you get to these high-level areas, that may no longer be a relevant boundary for that region. And so, you think of, there's really many, many boundaries, and I know obviously you've been thinking about this. And so how do you, come up with an approach that's systematic, that allows you to take into account all of the information that's available and all of the different boundaries, presumably, that are implemented across all of these different regions?
Well, I think that's a question about sort of the perspective you take. So like for us, we consider the boundary for any given area to be sort of the optimal boundary that we can get with a linear decoder. So when you ask like different areas, we wouldn't sort of kind of enforce one area's boundary on another. We'll assume that the brain knows from experience and training where the kind of boundary is between animate or inanimate or faces or non-faces. With respect to, I think, the second question, I think what you're getting at is something that we've been trying to sort of think about, and that what defines an object or a category is different in different regions, and it's quite possible the brain has the capacity to read out categories from different regions. So like the one example I think about is like Rufen van Roeland's group like a while ago had this studies like showing just the statistics of images of faces have certain regularities. And you can pull out those statistics in V1. So you can imagine if you were to sort of press an observer to be as fast, as accurate as possible, or let's just say as fast as possible, the brain might just say, well, good enough, let's go with V1, I'll be fast. And that would, like Simon Thorpe's work with the really fast saccades, might be consistent with that. It's not, maybe it doesn't mean that information gets all the way to IT. Maybe the brain is just saying, I'm going to use the best possible information to do this as fast as possible. I don't know if that really 100% got where you were. Sort of. Okay.
That was a beautiful talk, and I've definitely got decoder envy. But I wanted to sort of question, in a sense, what it is that's being modeled here, in the sense that you're assuming in effect, a static representational space. And one of the things that you you said was that you're not clear about why you'd want a sort of dynamic readout for objects. And I want to convince you that you need that, and that may explain maybe some of the messiness. So the way I think of the representational space, and many other people do, is there's some sort of experiential component to that. So things that can occur in similar kinds of contexts are represented more closely in this space. Where I think you need it to think of a more dynamic space is the kind of, well, there's an analogy in hearing research, which is that as I adapt to your accent, I'm shifting a little bit where I put my tokens. Okay, the vowel space, I hear your vowel, and now I'm going to put it somewhere else compared to hearing somebody else's voice. And I think it's the notion of token that's really important, and that's what defines the experience of an individual object. So here you're looking at some sort of generic kind of model, some sort of task, which actually is not a very common task in our everyday life. In our everyday life, we're actually experiencing specific objects that are tied to a specific episodic context. Mm-hmm. And that means that they're associated with all sorts of manner of things. And that association to the episodic context is what, in a sense, is in a local temporal way, redefining the semantic space. It's a little bit like pulling your attractors apart. Yep. And so one of the things that you showed at the very beginning was this sort of overlap, and the problem is, given this overlap in your representational space, how do you pull these things apart? Well, it may be that part of the answer for that is that actually in our experience of tokens of objects, instances that are experienced in an episodic context, that context is pulling these things apart. If one could model that, if one could take into account the dynamics of the space, dynamics across immediate experience rather than temporal unfolding as the signal passes through the brain, then maybe you'd be able to better pull apart these things, and these models might have a chance.
Yeah, I see your point. Let's see if we can go to one of these spaces. And I think it's a little, the problem that I'm talking about is a little different, but let's get to the target, non-target one. Yeah, here. So I think you're saying like there's a lot of imports of context and experience and object recognition. That I definitely accept that to be true, and I think few people would disagree. But I would think of that as sort of that contextual information being used to push these little individual dots around and push them sort of like away from the boundary. The problem that I'm describing with dynamic representations is there's two. So if we go to where's the baseline boundary problem? Yeah, here. So what that is basically for every time point, we're going to fit a classifier and train that. And of course, because there's sort of just noise there, that classifier would generate, or it would perform at chance. A dynamic model would basically say that, yeah, step one, it uses this decision boundary. Step two, it uses this decision boundary. The problem with that system is then the brain actually has to know which step it's in. And that could be, it could be signaled, that's possible. So I've had discussions about a hierarchical thing, like a new stimulus impinges on the retina, it resets the clock, and then it starts moving the decision boundary. And that's conceivable, but it's just a much more complex system. And there, in like the motor movement literature, there's definitely good research showing of dynamic state spaces and things like that. But I'm yet to be convinced that it's, at least now, kind of really relevant for parts of the model. As I said, we already have a whole bucket list of things to kick around, but it might be the case that dynamics will actually fix some of those things.
Thanks, Tom. That's so interesting. So Dave Leopold had a great poster last year at SFN where he showed even within these AF face patch, there's tremendous heterogeneity between different neurons of where they're connected to and what they respond to. And even in the area MT, where you know that neurons like direction, it's still interesting to think about, well, geez, if you're trying to decode direction, should you look at all the neurons, or just the neurons that like the preferred direction, or the neurons that like a little bit off preferred, because then you're on the sharp slope of the tuning curve? So can you talk a little bit about what that would mean for these? Because I hear you're just dumping in all the neurons, but maybe the brain is saying, hey, for this judgment, I only should listen to these 100 neurons and not these other 1,000 neurons. And if you're dumping all of them into your decoder, that might change your view of how the brain could be doing the task.
I think that's a great question. It's one that's going to be done on, it's also one to think about on multiple scales. So the first level is like, okay, so we have 30 neurons that we have. Maybe our classifier, or maybe the readout mechanism is only connected to 15, and how are we going to figure that out? I think that's an important problem in that it's kind of like the other one is we need to know what information is being used by an area to really sort of properly evaluate the classifier or the model. But you can scale that up too. Like our original study was done using Nico's data, which had all of inferior temporal cortex. And if you really think about that, you're like, well, is it possible that basically every neuron in inferior temporal cortex is all wired up to one decision process? Probably not. So then you have to think about, okay, what's the scale of the area that we need to be kind of looking at for readout? And part of your question, and we're working on this, is, and I love objects, but we've actually moved to looking at MT physiology data because with MT and motion, we have a better idea what ground truth is. In some of my talks, I have these slides like, where I ask these questions like, we don't know what the opposite of a giraffe is, but we know what the opposite of leftward motion is, and we know at different speeds, and we can parametrically evaluate move things. So I think in making progress on this readout, we'll probably be making more headway when we look at the MT data, just because we have a better understanding of the ground truth. Yeah. And even, I would say, with the MT data we looked at a lot of the methods that decoders use, and then particularly the exploratory methods, and a lot of them catastrophically fail to pull out basic motion features that can't unpack speed and direction, and when you just throw it into something that's going to do like a higher cluster analysis, which should give us all pause when we use these methods and to try to interpret this multivariate data.
Yeah, hi, Tom.
Oh my. I just had a little flashback. Sorry.
It's a terrific talk. So one of the take-home messages that I got from this was that a lot of these models will be successful, and that we have a problem with trying to adjudicate between which one is actually mirroring what the brain is actually doing. So along those lines, and along, I think, the issue that Jerry just brought up, linking it to behavior, well, links it to a particular context. So, the animate-inanimate kind of decision it's kind of a weird kind of thing to ask people to do, right? As opposed to having people name the objects. So I don't know if you've done that kind of task, but at least what is your thinking in terms of, let's say you had object naming data. Would that change the kind of answer you're getting, do you think?
I think you're definitely on the right track because object naming data is it would correspond much more to kind of our natural set of choices. And I believe somebody actually suggested it not for when we were working originally with the 92 objects, and we just didn't think we had enough power to do it. But we have not tried that with the neurophysiology data. I mean, but that brings up something that I also didn't really talk about. Like one of the issues with looking at the neurophysiology data, and I know I definitely skirted over it, but we're using human behavior to basically evaluate the models. So now a human is becoming the homunculus reading out from these areas. That said, I think I think your point is well taken. Naming data, I have to concede because otherwise, no. I think naming data is a much more straightforward way to go.
Right. So I was just trying to pin you down in terms of whether you think there's kind of an organization that is independent of task or not. You know, one way of thinking about this is that it's all context dependent versus this idea of a core representation that we could get to independent of what we're asking people to do, even though we need those data to do the adjudication, right?
Right.
So you're not...
Yeah.
Not scared of him. Not scared of me.
So to push a little bit more on what Jerry and Michael have been asking you, so I have the privilege of also having worked with the AF neurons. I'm actually recording from them right now using a slightly different task, which is looking at how they respond to more real-world stimuli like movies. And so the reason I want to bring this up is this context stuff that's going on that Jerry brought up and the dynamics and all that. We find actually, looking further at these neurons, that the response properties are extremely complex when you get to a more real-world stimulus. And you brought up earlier that, well, when the eye moves, you know, something new hits the retina, it just restarts the system. Yep. But we actually have found, especially in stuff like AF, way down the stream, that the movement of the eye is meaningless to what these cells are doing. They kind of ignore where the eye is, and they are ticking along doing the same thing trial by trial, regardless of the fact that the monkey does something different each trial, suggesting that the movement of the eye, the recentering of the fovea, is not actually influencing the response properties in these much later regions, and that something like the category selectivity of these cells kind of drops off drastically when you get to this more real-world behavior. And so the question kind of becomes, how useful is this feature space that we keep trying to map onto the brain when really the world is much more continuous and features are constant, not flashing at you? And so can we actually use these decision boundaries when there is no onset response for a lot of these areas?
I mean, in a way, what you're saying, I think I almost take as sort of good news. Because like one of the things that we see like in the single unit recordings is they'll sometimes there'll be a kind of a drop in the response. They'll kind of like have a dynamic where there's a dip and then a peak. And if you imagine that thinking about in terms of representational spaces, that would mean animates would flip to one side of the boundary and then go to the other. So it's good that you say in the natural environment they're sort of ticking along without and ignoring the eye movements and things like that. It makes me think that, well, there's a more stable system that we can work with. With regards to the natural images, I would definitely like to move like to that direction once we sort of have the models more developed. I do think it's good to start with a simplified approach. Yeah, and I would love to talk more about your data later. That'd be great.
Last two questions.
Not, okay. Excuse me. So I love that you're using the distances from these decision boundaries to explain this aspect of behavior. So this is probably not fair, but that makes me start thinking of, can you explain all aspects of behavior in these kind of choice tasks? So what I was thinking about when you were showing your data is, other things that affect response times or accuracy in these kinds of decision-making tasks, and whether, and I'll give you some examples in a second, but whether or not you think this distance from the boundary should be able to explain these effects, or whether you think there's some other mechanism driving them. And the two things that came to mind are related. So the first is like a semantic priming type effect. So we know we can make people faster or slower on making a living, non-living judgment by something they've done just before, or if they've seen that item a minute ago, even if they haven't done that decision. So the question is, what in this would have changed in a priming context to facilitate response time? And the second, kind of related, is thinking about judgments, like if you have people, classifying, a male face from a female face, and you show an adapter over and over again, a lot of male faces, it seems like you kind of move their boundary. So now you show them a female face and, along a continuum, and they seem to have a different cut point after an adapter for a while. And again, the question is something like that, what in this system would be changing to cause those two behavioral changes to the exact same stimulus? And that's what those two things have in common. So the stimulus is the same, the judgment is the same, but the response time or the accuracy is different.
Yeah, well, let's say there's two bits to that. I think that goes back to the dynamics thing. And I think we want to continue to assume the boundary doesn't move, because that's moving to a dynamic model. So what I would like to think with things like priming is you're pushing the dots around in the space. So if this is your exemplar here, right, with this dot here, the fact that it's preceded by a face might sort of, maybe you'll start in we are going to dynamics, but maybe the representation was already here because you just saw a face, and then you see another face and it gets pushed further from the boundary. I would say, but I'd also add.
How does that happen? Why is your response to it being pushed further from the boundary?
Okay, so like just think of it, we're in a state space, and we just saw a face, so then we would be on this side of the boundary already, and then you show another face, and it would push it further. Yeah, I know that's dynamics.
Okay.
But it's not the dynamics that I'm talking about in terms of moving. So I'm happy to concede that the dots will move around in the state space, and it's something that I've been playing around with for like two years. What I'm saying about dynamic models is then the decision boundary actually moves.
But if the dots move, doesn't the decision boundary move?
You'd have to have a model of how the decision boundary stays constant in the face of the shifting semantic space.
I'm not saying the space shifts, I'm saying the point shifts. So if we see John, it's a point here, and then we see another picture of Bob, that it pushes further away from the boundary. So the distance from the boundary gets larger, and then you're faster to react, but the boundary still stays in the same location.
Thanks. So there have been a lot of comments about the importance of external or, I guess, environmental context. But one thing I guess that I haven't heard explicitly brought up is the importance of internal context. So, in your model as you presented it, and perhaps this is a caricature, there's some part of the brain that's reading out the information specified in the inferior temporal cortex. But that sort of doesn't account for or ignores the possibility from feedback from other decision-making parts of the brain onto the decoding of information in the inferior temporal cortex. So we all know of these examples like, looking at a rock on Mars and perceiving it as a face. I mean, that tells you something about your predisposition to interpret visual information in one way or another, and certainly that must have an effect, if not on where a boundary is, then on how you classify an object with respect to the boundary. So I think that I wonder if, in other words, the information, I guess, visual information in the inferior temporal cortex underspecifies this, or the decision is underspecified by that information, and you have to take into account the the internal contextual state and feedback from other cortical areas.
Yeah, I think that follows the trajectory we've been talking a lot. And it really, like I say, my view is that is sort of pushing little individuals around the space as opposed to moving the boundary. And again, I could be wrong about moving the boundary. It just makes for a much more complex system. And it's not that I'm necessarily afraid of that system. It's just I haven't found good reason to move to that level. It's easier to think of in terms of moving because maybe a face on the moon is just something close to the boundary, and maybe because they just saw a face that pushes it even further.
Well, I wonder, just as a small last point, I wonder if these kinds of effects of salience to the system that is performing the readout might explain some of the inconsistencies in your data, like the fact that that distance from boundary determines decision making for animate but not inanimate objects. Maybe animate objects are just more important, so you want to be really careful about making that decision, whereas an inanimate object, as soon as you recognize that it really is inanimate, it just doesn't matter anymore.
I think that goes, well, it sort of flips around a little bit to Alex's point as well, it is an unnatural task. First, the animate, inanimate is very unnatural, even though it's a big distinction in IT. We don't walk around day going animate, inanimate, inanimate. Do you? We might want to study you. But there's also the problem of negative categories, which is something we thought about, and that animate is a category of objects. Inanimate is the absence of those features. But also with the inanimates, one of the things that we've encountered, so we have gotten the effects for inanimate objects in certain data, including the monkey data. And part of the problem is the variance for inanimate objects reaction times is really small, while the variance for animate objects is very large. And because we just don't have enough variance, might be one of the reasons why the inanimate doesn't work. The fact that it's a negatively defined category is another reason that it does work. Or we might need a better model.
But that goes to the importance of context.
He's shutting you down. Thank you. Thank you. Thank you, everyone.